disease is sort of what spawned the field of genomics. I was actually studying neurotransmitter receptors. So in '84, I joined the National Institutes of Health and sequenced the first adrenaline receptor from the human brain, and using the standard gel and radioactive technologies, it took over a year to do this one gene. It was painful. It was, you know, good to guess what the sequences actually were from these gels, and it's also right around when Lee Hood's lab announced that they had this new method of attaching 4 different colored dyes to the bases of DNA and could activate them with a light beam and read that into a computer as it ran down a gel. And so we were my lab at NIH was actually the first one to get this working. In fact, in '87, I think we published the first paper sequencing genes with this technology. But evolutionary biology was moving very slowly, and you sort of did one gene from different species. And so it was around '85. In fact, the Genome Project started with a suggestion here in La Jolla. And you know, I just got immediately excited about it. The notion of sequencing every gene versus the way they were being done one at a time, usually a decade per gene per laboratory, and it was a rough guesstimation. So the rate science was proceeding without genomics, we'd have maybe a quarter of the human genes discovered by now. Maybe not. It was going to be a long, long road because they were done as I was approaching adrenaline with a function that you could trace down and then try and get the gene associated with it. And I just got excited about it, and having the only lab that had an automated sequencer working, you know, I thought, gee, we have the technology that could actually do this. Jim Watson got very excited about it, and it seems pretty ludicrous today when we look back at these early machines. It's like trying to do a 100-mile bicycle race on a unicycle with a flat tire. I mean, they barely sequenced 16 reads per day. But you know, the notion was, you know, as it happened with Henry Ford and you know the whole manufacturing scenario, one's not enough. You buy a whole bunch. And it led to, in '87, being annoyed with the slow pace that the government was moving. It wouldn't have finished even this century at the rate it was going. So Applied Biosystems introduced its version of the Catlair sequencer, which actually worked. That's why I'm in the middle there. But I've changed now. They've improved. So we bought 350 of these and set up a giant factory, and that led to the first draft of the human genome. And you know, but it's just getting started. In part, you know, the only thing that's really changed in the last 15 years since we sequenced the first draft is the technology, and it is really stunning. So we had this building with 350 of these instruments. It took 9 months to sequence the first draft of the human genome. And the technology has progressed. You know, Illumina in particular has, with their announcement of the X10 sequencer, has a machine that I wish the hell I had 15 years ago. That we're scaling up now with 2 of these units to do 40,000 genomes a year versus 1 in that same time frame. And you know, Life has developed some exciting technologies as well, and you know, that's the value of the San Diego community. It's now the home of this entire industry. And right after we announced HLI, I got a call from a Boston Globe reporter saying, "So you're telling me the center of genomics has now moved to La Jolla?" And I said, "You're just recognizing it."
Yeah, they're slow to come.
Yeah,
They're in denial.
They're in denial for a while.